This is the parameter form of minimap2 in Galaxy. It has the typical parameters where you define the reference genome and the FASTQ files that you want to use as input. It has a long list of additional parameters that are divided into 4 groups, as you can see here. And this comes back in the slides. So the groups have gotten a separate color. So when you see an option that's colored red, it means that you have to search for these parameters in the indexing options section here in Galaxy. So you have to expand this and then you will find that parameter. So Minimap starts by chopping the reference genome into smaller chunks so that they fit in the memory of your computer, and the size of the chunks is defined by the -i parameter that is translated in Galaxy into this one here, split index for every n input gigabases. So in the original documentation, this is the -i parameter. So if you have a computer that doesn't have a lot of memory, you can use this parameter to specify the amount of memory that the process can take. There's also the -h parameter, which is related to compressing homopolymers. So homopolymers are stretches of identical bases, like you see here. So you can also find that parameter here in the indexing options. If you enable the compression of homopolymers, then this homopolymer will be compressed into a single A by Minimap. This is done because sequencing these homopolymers is very tricky. Long-read sequencers like PacBio and Nanopore often read homopolymers as a single nucleotide. So this is exactly the behavior that they show. Short-read sequencers like Illumina will show the opposite behavior. So when they encounter a homopolymer, they will often add an extra base. So AA will become AAA. So for short reads, this parameter should be kept at no. And that's exactly what the default value is, as you can see here. So Minimap will cut the chunks of reference genome into small seeds, and in Galaxy the seeds are called minimizers. And that's what you see here. These seeds have a certain length that is specified here by this parameter, so in the original documentation -k. In Galaxy it's called the Kamer size, and the default setting is 16. Now I'll show the example for a much smaller setting. So let's take seeds with length 4. So this is why you see these small sequences here that are all 4 bases long. It's because I chose a seed length that was equal to 4. You not only have to specify the length of the seeds, you also have to specify the size of the window that you're going to use, and this is done originally by the -w parameter, but in Galaxy this is translated into the minimizer window size. So, and the default here is 10. And this is why you see these seeds being grouped in groups of 5, because I'm choosing here in the example for a window size of 5. So what will he do? This is your reference genome sequence. So he will go through the reference genome and he will take small seeds, so small subsequences with a length of 4. So the first one is AGCA. And then he will move one position and he will take a new seed. So that's what you see here. He will move another position and he will take a new seed and so on. And he will do this 5 times because the window size is 5. So you will get this group of seeds. And then he chooses the seed that comes first in the alphabet, which is in this case this one. Then he moves the window one position, so he will start here now and will produce 5 seeds of length 4. And in that group of 5 seeds, he will determine which one comes first in the alphabet. So in this case, it's this one. He will move the window again one position. He will produce 5 seeds of length 4 and decide which one comes first in the alphabet. It's still this one. And this is how he picks a seed for every window that he looks at. Now, with these seeds, he's going to build a table that looks like this. It will contain the sequences of the seeds and their positions in the genome. And with positions, I mean the number of the window that they were selected in. So this seed will have position 1, and this seed will have position 2 and 3. But consecutive positions are not saved, so for this seed he will only save position 2.